Hey, Hammy here, back with the last video, section 2.3, part 2 of chapter 2. Woo! Okay, and then we're done with biochemistry and we can move on to cells. But before we wrap up this chapter, we need to talk a little bit about acids and bases and the pH scale. First, a little review from physical science last year. Okay, when you put two things together, that makes a mixture, and you talked about different kinds of mixtures. Okay, in particular, a solution is a mixture that has a similar, equal composition throughout. It's equally mixed all through the solution. Okay, acids and bases only happen in solution. Okay, pure water, which is H2O. Okay, so remember, this is our oxygen, and these are our two hydrogens. When you have just a beaker of pure water sitting there, some of the water molecules will dissociate. Okay, dissociate or break into two. Okay, they break into two ions. Okay, the first one here, and actually this is flipped as opposed to what it is in your notes. The first one here, the O is a OH ion. Okay, so one of the hydrogens here will jump and move over to this water molecule over here. Okay, so the one that lost a hydrogen ion is known as a hydroxide ion, and it has a negative charge. Okay, negative charge. The other water molecule that gained the hydrogen is known as a hydronium ion. Okay, and it has a +1 charge. Okay, so water is constantly dissociating or breaking into these two things. Now notice there's also an arrow going back, reversible. So while some of the water molecules are breaking apart, some of the water molecules over here are going back together. That hydrogen is jumping back, and you have two just plain H2Os again. And so this is constantly happening throughout your beaker of water, kind of equally all throughout the water. Okay, sometimes when you add substances to water, they don't break equally, or it makes more hydronium, or sometimes some books back in my day we used to call them as H pluses. And this, like extra hydrogens that are leaving a water molecule, and things that do this are called acids. So this increases the acidity of the solution. And we measure that on the pH scale. So down here is an example of the pH scale with giving you some examples of common items. The pH scale ranges from 0 to 14. Okay, with 0 to 7, okay, this region right here, all being what? Acids, right? 0 to 7 are known as acids, okay, or it's the acidic end of the pH scale. The pH of 7 right here is neutral, and from 7 or above 7 to 14, okay, these things are known as, sometimes you hear basic, or like on the chart down here, another term is alkaline. Okay, so you can see pure water, pure water has a pH of 7 or is neutral. Okay, things that tend to be alkaline or basic, think of like cleaners, soap, laundry detergent, lye, sodium hydroxide, is very basic and can actually cause burns on the skin. They tend to have kind of a slippery feel, and if you've ever had your mouth washed out with soap, it has kind of a bitter taste. Whereas acids, we see a lot of things that we eat and drink over here that are acidic. Acids tend to taste sour, have a sour taste, like lemon juice. Citric acid is an acid. Okay. Also notice that if we go down from 7 down to 6, okay, from blood down to mineral water, one step would be a 10 times change 10 times the amount of hydrogen or H plus ions or hydronium ions in the solution. If we go another step, let's say we go from mineral water at 6 to wine down to 4, that's 2 steps. Okay, that'd be 10 times 10 or 100 times more acidic. Okay, so you understand that this is a logarithmic scale and each step is 10 times more than the next, so you have to multiply the two. Finally, why important to living things? Well, acids and bases, especially when it comes to enzymes, happen in different places of our body. If we look at 3 enzymes here, pepsin, salivary amylase, and trypsin. Pepsin and trypsin break down proteins, pepsin, trypsin. Salivary, like saliva, breaks down starch in our mouth. Now notice they all work, they have peak activities at different pH levels. At about 7, this works at about 9, pepsin works at about 2. Okay, why is that? Well, salivary amylase, saliva, starch digestion starts in your mouth. That enzyme works best at about a neutral pH, which would be inside your mouth, your saliva. Okay, when you swallow your food, trypsin begins to digest proteins, pepsin begins to digest proteins because it has a pH of 2. This is found in your stomach, and stomach acid, hydrochloric acid, okay, there are the H plus ions that'll be released, makes it very acidic, so pepsin can only work in a very acidic environment. When your food is kind of squirted as chyme, C-H-Y-M, into the small intestine, the small intestine adds sodium bicarbonate ions into it and raises the pH level actually above neutral a little bit to a pH of about 8 or 9. So trypsin continues protein digestion then all through your small intestine, but it works better at a higher pH because you can't have this acid going all through your intestines. The acid is just in your stomach. Okay, so in different living things, different environments, enzymes, which we talked about in previous videos, for all those chemical reactions to take place, metabolism, we use that term, they need to work in different pHs. And so our cells have different enzymes that work in different pHs, or they get denatured and don't work. So I hope that gives you a little better understanding of what makes something an acid, what makes something a base, and the pH scale that we use to measure it.